Thanks so much for the kind introduction and, of course, the possibility to speak at today's event. I had a really fabulous morning and I enjoyed the talks. So now I invite you to think about chemistry. This is slightly different what we have heard so far. We're a group of chemists based at the Crick and King's College London, and we use metal complexes as potential or as probes and as potential therapeutics in biological systems. And of course, for today's context, this is in the field of AMR. But I thought I'd get started by really showing you the breadth of what we are doing in my research group. So it's all around metal complexes, but we have different research areas. So in I love it, the Shape It Up project, we basically integrate metal complexes with traditional drug discovery programs, such as fragment-based drug discovery, to really enable the rational design of metal complexes and going away from just making compounds and phenotypically screen them. So we have now developed a high-throughput screening approach where we can virtually screen compounds and then design them as highly potent enzyme inhibitors. We're also using metal complex as catalysts. So for the chemist in the room, you're well aware metal complexes are used for your chemical transformations. And I want to show you how we can use metal catalysis in biological systems today. And I would argue metal complexes have also the capabilities of being exceptional scaffolds for drug discovery because they can simultaneously bridge DNA and proteins, and we have shown that they are actually very potent DNA gyrase and topo4 inhibitors. So watch out the space. There is a preprint. Hopefully I will submit this on Monday, so you will hear from us soon about this. And then, of course, there is also a lot of work going into technology development, and this is with my colleagues at the Crick, where we really want to understand what compounds do actually accumulate in gram-negatives. We all know this is a really essential factor if you want to design an effective antibiotics. And then, in the end, if we know which compounds or which metal complex do accumulate in bacteria, we have worked with GSK to show that we can use chemoproteomics approaches to really pinpoint the targets of our metal complexes. So let me just give you a really brief brush up on metals in medicine. So this is nothing new. Metals are used in medicine to date and have been for many years. Maybe you're familiar and have seen this compound before, right? Cisplatin is used in almost 50% of all cancer patients today. This is a fairly old example, but just to show you, there are also other metal-based compounds in the clinic which are based on gold or silver. And more recently, we have seen these ruthenium-based compounds entering clinical trials for the treatment of cancer. So there's clearly an appetite that we diversify and open the chemical space. But really the question for us is, why do we actually want to use metal? So metal complexes have the capability of accessing multiple mechanisms of actions at the same time. And if we want to address resistance, this is really interesting for us in the hope that maybe bacteria won't evolve resistance to multi or polypharmacology. So metal complexes can undergo ligand exchange reaction. They can bind to DNA. We can design them that they produce reactive oxygen species. And as said, they are catalysts, and we can use them catalytically in biological systems. And here at the center, basically I want to illustrate to you that metal complexes have a huge structural diversity. They are like an extended Lego set to your basic Lego set, where you can then build metal complexes in all different shapes and form, from octahedral to piano stool complexes, which you can't easily access with purely organic compounds. So But let me start first and showing you here a little example of what we have done recently in our efforts of designing catalysts that work in biological systems. And this was really driven by the need of having new drug modalities that address AMR. And you may be familiar in how PROTACs work, right? They modulate the disease by targeted protein degradation. You have here your compound, the PROTAC, which consists of a heterobifunctional molecule. You have a binder for your protein of interest, and you have an E3 ligase binder. And then your PROTAC brings these two proteins together, forming the ternary complex, enabling ubiquitination and ultimately proteasomal degradation. And we have seen that this is a very successful approach in oncology, but unfortunately that doesn't work in bacteria because we don't have E3 ligases. So my colleague Esther Moreale is working on the development of BAC-PROTAC and have shown that she can recruit the bacterial proteasomal degradation, such as ClIP-CP, in gram-positives and microbacteria. However, so far we could not degrade proteins in gram-negatives, and this is what my group tried to address. And here we basically got inspiration from photodynamic therapy. This is a technique where metals are already used in cancer therapy. And to put this kind of complicated diagram in simple terms, the metal complex can serve as a photosensitizer. You shine light on it, it gets to an excited state, and then it can react with biomolecules to produce reactive oxygen species, which are lethal. So now let's bring all these concepts together. We were aimed at designing and enabling targeted protein degradation mediated by light-activated metal complexes. So here we have our molecule. It's still a heterobifunctional molecule, but this time we have our organic directing group specific for our protein of interest, and we completely substituted the bacterial proteasomal degradation system with a metal complex. So our organic molecule will bring our metal complex to our protein of interest, and here we have an extra trigger because we can decide when and where we shine light, and only there our metal complex will produce reactive oxygen species, will damage our protein, will release our molecule, and we can enter a new catalytic cycle. Well, that was the theory. Does this actually work? So in a proof of concept study, we tried to address the problem of resistance to beta-lactams and the expression of beta-lactamases. And we already know that combination therapies of beta-lactam and serine beta-lactamases are used in the clinic, but there are no clinically approved metallo-beta-lactamases. So we really wanted to fill this gap, and therefore were aimed at targeting the New Delhi metallo-beta-lactamase 1, which belongs to this class. So the first question was, can we actually find a binder to NDM1? And we looked into the literature, and Chen et al. Reported in 2017, and J. Metcalm, this dipicolinic acid organic compound which would inhibit NDM1. So we have attached N1 to our ruthenium photosensitizer, generating our heterobifunctional molecule Ru1. And of course, you always need a control, here Ru2, which does not have this targeting unit. So as a first step, very briefly, just to show you, we had to assess if actually these compounds do produce reactive oxygen species. So these are quite standard excitation and emission spectra, what we would expect. We also showed that the lifetime of the triplet excited state, which is really essential for this process to happen, is long-lived. And we also did EPR studies to really show what type of reactive oxygen species we produce. And you can see both Ru1 and Ru2 produce reactive oxygen species. These are the triplet signals of singlet oxygen, and we determined this with EPR. Happy to discuss the details later on in the coffee break if you want. But basically we're all set to go. And so the next step was, can we now also inhibit NDM1? And for this we recombinantly expressed and purified NDM1 and used this nitrocephin assay, which is a calorimetric assay where you see inhibition when you basically the color changes from yellow to orange if the enzyme is active. So we titrated in our Ru1 complex in the absence of light, and we saw this is indeed an inhibitor at around 23 micromolar. But now when we have a single flash of light, we see that we have 100-fold improvement in its inhibition down to 220 nanomolar. So what happens with Ru2? In the absence of light, this compound is not an inhibitor of NDM1, but when we shine light, these compounds will produce ROS. We know this from our photophysical characterization, and we have an IC50 of around 5 micromolar. But this gives us a handle to understand what is the selectivity in our in vitro system, which is around 25-fold if you have this targeting unit. So in the next step, we wanted to see how selective is this process with Ru1 when we mix NDM1 with another protein, such as BSA. And this is the experiment in the absence of light at various concentration of our compound. And you see none of the proteins are affected. We also have here a nice mass spec. But when we now shine light, we can see that we selectively and exclusively only damage NDM1 and not BSA, so we leave this unaffected. So that was great, and we wanted to move now to live bacteria, but for this we had to assess if our compounds are accumulated. I told you, and I guess you're all aware, it's extremely challenging to get compounds into gram-negatives. And here is another beauty of metals and why we love them. They're inherently phosphorescent, so we can directly follow their uptake by confocal microscopy. So you see here Ru1 is accumulating in E. coli, and we have also control, HADA, which stains the peptidoglycan. So that was like really nice. Our compound gets into the cytoplasm. And we also used an alternative technique, which is based on LCMS, where we showed all of our compounds do accumulate, so we can use them as controls, and they accumulate in both NDM1 and empty E. coli strains. So then the next step was to adapt our nitrocephin assay to NDM1 E. coli strain. And basically here you see that when we don't shine light and we use again our targeted compound, we see the same inhibition of NDM1 by our compound. And when we irradiate the system with light, we see the effect of the inhibition of NDM1 in E. coli of around 750 nanomolar. But I think really important here is that our untargeted complex, while we saw activity in the in vitro assay in the live bacteria with an untargeted complex, we don't see any inhibition of NDM1. So clearly the targeting unit is extremely important. But then, of course, next, can we now actually rescue the activity of meropenem? And for this, we had to assess the MICs, and you can see these compounds are not antibiotics themselves. They actually serve as adjuvants. And you can see also here the difference of an E. coli empty strain and an NDM1 expressing strain where meropenem loses its activity. So now to see if these compounds can work in combination with meropenem, we did some checkerboard assays where we combined meropenem with our compounds in the absence and in the presence of light. And to basically put these nice figures into a table makes it nicely digestible. So just to show you that our Ru1 complex in the presence of light outperformed the all of the compounds, but at the highest concentration tested, we actually saw a 50-fold rescue of the meropenem activity. And with this, I just want to show you a brief summary of that we can indeed degrade proteins and damage protein in E. coli using metal complexes. We can show that these are selective processes, they accumulate, and they can rescue the activity of meropenem. And this is, for me, the most important slide. Nothing of this work would be possible without my dedicated group. It's really a pleasure to work with them. And I presented today work by my first PhD student, Lars, who's now a postdoc in the group. He was supported by Tom. I'm very grateful to all the STP support at the Crick, my collaborators, and of course the funding. Thanks for your attention, and I'm happy to take questions.